Hi everybody, it is 6:40 p.m. in Europe, in Brescia. I am talking from Brescia, which is one of the co-hosting university, and together with Dr. Christine Austin, I chair this session, and so you're welcome. We have 7 speakers today. You have about 4 minutes presentation and then some time for discussion. Then at the end, we can have a panel discussion. So I give the floor first to Dominic Brown, correct?
Yes.
So the floor is yours.
Thank you for introducing me and just minimizing everything here. Full screen, okay. So welcome to my today's talk about longitudinal investigation of infants' exposure to food contaminants via breast milk. And we jump right in. So infants are also exposed to environmental exposures because they're exclusively breastfed with breast milk. And obviously, if the mother is exposed to environmental exposures, the baby might be exposed to as well. And we wanted to investigate that in detail by developing multi-class biomonitoring methods. And we used a catchers-based extraction, so some catchers, salts, and we extracted our targeted analytes. And with the first method, we investigated xenobiotics from the supernatant. And with the second method, we applied a solid phase extraction and a concentration factor and measured mycotoxins. And we measured those with an LC-MS-MS, so a Q-trap system. And I want to focus on some results here. So you can see on the right-hand panel here, the altanariol monomethyl ether concentrations or mycotoxin. And what you can see on the x-axis is the delivery time point here on the left-hand side. And we sampled 87 samples from one mother throughout basically 6 months. And you can see the longitudinal exposure to this mycotoxin. And at 2 distinct days, here at day 26, and for example, here at day 166, the exposure was higher compared to the chronic low-dose exposure we saw the rest of the time. Here in this case, it was about 35 nanograms per liter, and here nearly 60 nanograms per liter. We also found other mycotoxins. These are plotted on the left-hand axis, and you can see that they're mostly below 10 nanograms per liter. And if we focus, for example, on the crossed circle, which is bovaricin, you can see that it's stable over time. So we also have here a chronic low-dose exposure. We also used these samples from this mother to investigate it with the second method we had. For xenobiotics, and we tested for different xenobiotics which are in personal care products or in plastic components or industrial chemicals or natural xenobiotics which are basically food contaminants or also food processing carcinogens. And you can see here some of the peaks, for example, some herbal tea components. We have here some other food processing carcinogens or also mycotoxins again, and some industrial chemicals, which we found in these 87 samples. So it was quite interesting to see these exposures for the exclusively breastfed infant. And the take-home message was that we found mainly lipophilic mycotoxins. Obviously, breast milk is a mostly lipid-based matrix. And we also found 29 xenobiotic compounds, so from industrial chemicals, but also herbal tea components, but mostly in the lower nanogram per liter range. We want to use these 2 methods we established to go into larger biomonitoring studies. Obviously, this was just one mother, so we want to use it in large screening studies. And with this, I really want to thank you and all my cooperation partners And yeah, if you have any questions, I put everything together in one slide here. So please ask.
Thank you so much, Dr. Braun. That was a very interesting talk. I mean, it must have been a lot of work to get that kind of resolution from one person, you know, so many days over 6 months. Please, if anyone's got any questions, please submit them via the chat, and Dr. Ranzi and I can forward your question on to the speaker. I'll get the ball rolling, Dr. Braun. Were you able to get, so there were a few, were you always collecting the milk at the same time? Or is there any way that you can look at whether the time you collected the milk, the exposures change?
You mean by timing every day at 8 in the morning?
Mm-hmm.
No, this is not possible because of ethical issues. So we really have to make sure that the baby is breastfed first before we sample. And sometimes it's not possible to sample because the baby basically empties the breast more or less completely. So you won't receive a sample, which can be donated. So most of the time it was more like a pooled sample over the day with the leftovers we had. And that's why also some data points are missing here as well. As you see, some we have not sampled because the baby basically was breastfed completely and we didn't receive any sample on these days.
Thank you.
You're welcome.
So there are any questions from the chat? It seems not. So we move to the next speaker, who is Paola Monti.
Okay. Good morning or good evening. Now I'm going to share screen. Okay. Thanks for this opportunity. The study that I'm going to present is focused on the effects of particulate matter exposure on the methylation of clock genes in a population of subjects with obesity or overweight. As you all know, particulate matter has been associated to many pathological conditions, and one of the principal pathological mechanisms started by PM consists in triggering a systemic inflammatory cascade. In this context, obesity and overweight could be hypersusceptibility factors to the noxious effects elicited by PM, since these conditions are characterized by a chronic low-grade inflammation. As recent studies have reported that particulate matter can affect DNA methylation, we first wanted to determine whether there is an association between particulate matter exposure and methylation of core clock genes listed here, which are the genes that control circadian rhythmicity in subjects with obesity or overweight. And second, we investigated the possible role of the body mass index of the BMI in the association between PM exposure and the methylation of clock genes. To achieve these aims, we recruited 200 women with obesity or overweight who were asked to donate a blood sample. After DNA extraction, we performed bisulfite pyrosequencing to determine the methylation level of core clock genes. Particulate matter exposure was assessed by monitoring stations, and both exposure data and the methylation data were put together to perform a statistical analysis. As a result, concerning particulate matter 10, we found that PM10 increments were associated to the hypermethylation of the clock, CRY1, CRY2, and PER3 genes, whereas for the PER2 gene, we observed an hypermethylation in the promoter region. Then we evaluated particulate matter 2.5, and we found that PM increments were associated to the hypermethylation of the CRY2 gene, whereas as the PM2.5 increases, we found that the promoter region of PER2 was hypermethylated. Finally, we evaluated the potential effect of the BMI on the association between particulate matter exposure and methylation of clock genes. And interestingly, we found that the BMI plays the role of an effect modifier for the PER1 gene. Indeed, for relatively low BMI values corresponding to 25, we found that the promoter region of PER1 was hypermethylated for both PM10 and PM2.5, whereas for very high BMI values corresponding to 51, we found that the promoter region was hypomethylated. So we conclude that particulate matter exposure can affect the methylation of clock genes in a population of subjects with obesity or overweight, and that the BMI could play the role of an effect modifier in the association between PM exposure and PER1 methylation. Further studies will be necessary to determine whether these methylation changes could result in alterations in gene expression, and if and how these changes could affect both the energetic metabolism and the endogenous clockwork. So thank you for your attention, and if you have any questions, feel free to ask or you can contact me. Thank you very much.
Is there any question also from the audience? You can chat your question. No question. Yes.
Sorry, I've got one question, and that's could exposure to PM 2.5 or PM 10, could that actually also be related to daylight cycles?
Yes, of course. We first we measure DNA, the DNA methylation level from blood samples which were collected at defined times in the morning to avoid possible confounders linked to natural variations in DNA methylation level. And of course, also PM values varies along the day time. So we took into consideration this feature in our statistical analysis. So yes, thank you for the question.
Thank you.
So thank you. Keep the time. We move to the next speakers. Then at the end, there is time for further questions. So the next one is Chrysobalantou Chatsioannou from Lyon.
Yes, hello, can you hear me?
Yes.
Yes. And I think you can also see my screen.
Yes, yes. Perfect.
Great. Welcome everybody to my presentation entitled Exploring the Dark Side of the Exposome. As it happens with many yet unexplored areas of science and life, while studying the exposome, certain risks are well understood, but a big part of the risk factors related to chronic disease mortality remain unknown. In order to improve our understanding, we applied an agnostic approach which could provide an overview of compounds not typically measured in targeted assays that usually are used for environmental studies. Trying to answer the question of how much could untargeted MS metabolomics help in the exposome understanding, we want to cover in a single run known exposures, but simultaneously discover new ones. To perform this study, we used samples from 2 epidemiological studies nested in the European Prospective Investigation into Cancer and Nutrition, known as EPIC cohort, and we applied the well-established protocol used in IARC for untargeted studies in reverse phase chromatography and both polarities. A list of 563 compounds was compiled to be used for suspect screening, including known diet and microbiome-related compounds, contaminants and pesticides, drugs and endogenous metabolites. An extensive list of standards which were analyzed in IARC were employed to achieve high confidence identification, as mentioned by previous speakers, preferably at level 1, meaning to meet the criteria of matching the theoretical monoisotopic exact mass, retention time, isotopic profile, and fragmentation spectra with commercial standards. Here is presented the chemical similarity map of 166 metabolites, which we were able to identify as level 1 or level 2 confidence with a Tanimoto coefficients higher than 0.7. Each dot of this plot represents a metabolite, and the size of the dots is proportional to the frequency of detection. So by comparing the pink and yellow dots, for instance, it is obvious that metabolites related to diet are much more frequently found in the samples analyzed compared to pharmaceuticals. An example of diet-related compounds detected and identified are low-calorie sweeteners that are used as sugar substitutes in the diet. Saccharin and A-sulfame K that are highlighted in the map could play a role of an objective biomarker of dietary intake instead of using the 24-hour recall questionnaires. For pharmaceuticals now, we observed that not only we were able to detect some parent compounds in low frequency, but also some of their metabolites based on highly correlated features. Such example is highlighted on the map with salicylic acid and its metabolite. An interesting observation is the high frequency of subpollutants such as propylparaben, and this compound was detected in the blood samples, but not in the black samples that we used in the analysis. On the contrary, other frequently detected pollutants such as triethyl phosphate were also detected in blanks. And this raises an alarm about the criteria of reporting a compound in such agnostic studies, where it is not feasible to take specific precautions for certain classes of compounds. So to conclude, we were able to detect a broad set of environmental compounds in blood through high-resolution MS metabolomics. Highly confidence identifications are crucial, and for that purpose, MS data are essential. Blanks are very important when studying contaminants, since certain compounds may be found everywhere, such as plasticizers. And interestingly, some metabolites of certain compounds can be monitored and are highly correlated with the parent compound. And closing, we should keep in mind that the detection of environmental compounds in blood is a matter of the exposure, but also of the limitations of the instrumentation that we are using. I would like to thank all colleagues and investigators that contributed to the study, and to thank you all for your kind attention. Thank you very much.
Yes, I see a question from the audience. How did you assess the biotransformation products?
So what we try to do is try to correlate some figures, some features that we find that sometimes they have, or we believe that they have a certain group in common with a compound, and we see that they are highly correlated. But this, we observe that it doesn't happen. So as an example, I gave the salicylic acid, but for instance, it happens that with acetyl salicylic acid, we don't see correlation. So this needs further to be investigated, because it can be that such compounds come from other sources also, and not only from pharmaceuticals, such as this example that I gave.
So thank you. Any other question? No. Okay, so we move to the next speaker. Christine, can you help me? Who is the next one? Marta Gallego Pauls, correct?
Yes.
Yes.
Okay. The floor is yours.
Okay, so I'm Marta. I'm a PhD student at the Barcelona Institute for Global Health, and I'm going to present my study on the variability of multi-omics profiles in a population-based child cohort. Right. So the early life is one of the most sensitive and vulnerable periods to environmental exposures, and by integrating multiple omics profiles, we are able to capture very early and subtle molecular responses to these, and therefore obtain a holistic view of the whole molecular response. However, there is an urgent need to study variability of omics profiles because exposome studies need to be informed of it in order to interpret changes. This is why the aims of our study were to estimate the variability between and within individuals and between the cohorts in our study, and to assess interrelationships across omics through a network analysis. We further decomposed variants according to several explanatory variables, although the results of this part will not be presented here today. So we studied 156 children from 5 European countries that took part of the ELIX panel study, and we collected blood and urine samples in 2 visits, 6 months apart. From blood, we measured DNA methylation, gene expression, microRNA, proteins, and serum metabolites, and also urine metabolites. We performed a variance partition analysis using linear mixed effect models in order to partition variants according to intra-individual, inter-individual, and cohort variability. And we also performed a network analysis using Gaussian graphical models based on the change between visit of the DNA methylation, the proteins, and serum and urine metabolites. So the results of the variance partition analysis showed large heterogeneity within and between omics. Features behave very differently even inside the same omics layer. However, all of the omics profiles presented a higher median intra-individual variability. We found that DNA methylation was the most stable omic profile because it had the highest inter-individual variability. And we could see also that the metabolites, both serum and urine, were highly variable. This is the resulting network that we obtained. You can see that each node color represents an omics layer, and we didn't find many dependencies across omics layers, since you can see that most of the connected components are formed by features from the same layer, except for some metabolites that were related to certain CPGs. So this study offers a multi-omics perspective of the medium-term variability in childhood. We found strong variability within omics profiles. This information is very valuable for the interpretation of variability in the context of large epidemiological studies. And with our cross-omic approach using networks, we exploited omics data to its full potential for preventing and predicting disease. And we didn't find many interdependencies across omics layers. So to conclude, we provide a valuable framework for further epidemiological studies that aim to detect omics signatures linked to disease, environmental exposures, or both. So thank you for your attention. If you have any questions.
Thank you very much, Mara. That was very interesting. I think that highlights one of the major challenges of studying the exposome is that it's so variable depending on the matrix you're looking at, the techniques you're looking at. And even within an individual, it varies quite significantly. So I have a two-part question. One is, what's the age range of the children that you studied? And the second part of that question is, do you think the within individual variability would change depending on what age they are?
So for the first part, the children under study were between 6 and 8 years old. And regarding the age, age was actually one of the variables that we included in that part of the study, and we didn't find much percentage of the variance that depended on the age. But for example, in some omics, such as serum metabolome, the age was a little important, like 7% of the variability. So it's not very, very relevant, but it's true that age affects or accounts for some of the variability that we see in the omics.
Great, thank you. We have a question here from Michelle Heacock. Why do you think methylation was the most stable?
So methylation is actually considered by a previous study the most stable profile. And I think it's because methylation signatures persist over time. So methylation is a modification of the DNA. So their modifications are persistent over time, and they are not as immediate as, for example, the metabolites, whose variation can be daily. Methylation is a modification like more persistent over time.
Thank you.
Thank you, Marta. So we move to the next speaker, who is Peng Gao, Stanford University. Hi.
Hello, everyone. Thank you very much, Dr. Ranzi, for your introduction. Today I'm going to talk about precision environmental health monitoring by longitudinal exposome and multi-omics profiling. The reason why we are interested in the precision environmental health is because the conventional way to monitor the environmental health is using environmental epidemiology studies. However, a few studies recently have found that even they monitor the individual living in the same city or same area, they have totally different exposure. And we didn't do too much study on monitoring the exposome or environmental health on personal level. So that's the reason we did this study. The way that we monitor the precision environmental health is to monitor the personal exposome and the profiling the multi-omics during the same period. This is the overview of our study. We have a device called the exposometer. It's a device that can absorb the airborne exposome longitudinally with the participant. And the device can collect the data of the environmental factors, and as well as there are two filters in the exposometer. One can collect the biological stressors, the other can collect the chemical stressors. We also collect the stool sample and the blood sample during the same period. For the stool sample, we did a 16S rRNA analysis for the bacteria. For the blood sample, we did a cytokine and a blood test as a traditional clinical measurement to reflect the human health. The other three omics are toxin and carcinogens. Also, AKS blood exposome that we considered before. The rest two are metabolome and proteome that are frequently studied before. And we were able to annotate like 158 compounds. Most of them are carcinogens or toxins from 3,000-ish LCMS features. And for the biological substance, we have like 1,265 genera. And as I mentioned, our goal is to investigate the combined effect of all the stressors, not a single group of them. And we all know that the environmental exposure are low doses and chronic. So we want to know the long-term effect of all the stressors. There are three kind of specific aim of our study. We want to measure stressors as much as possible. That's the reason we measure three different kind of groups. And also we want to know the comprehensive impact of the exposure on human health. So we did multi-omics integrations. The last one is we want to identify the health-related stressors by correlated the blood test and cytokine with the exposome. Here is the method we use. We recently published on the Nature Protocols. Briefly, for the chemical exposome, we used the LCMS, and for the biological exposome, we used next generation sequencing. Here is the overview of the physical stressors, biological stressors, and chemical stressors we did determine in these studies. And this order is from the low concentration to the high concentration. And we found that some pesticides, which are also agrochemicals and pharmaceuticals and personal care products, PPCPs, and group 2 carcinogens decreased during the periods. And also some other kind of pesticides and PPCPs and group 2 carcinogens increased. And I don't have a table for the details right now, but if you are interested, you can contact me later. And for the biological substance, we found there is a fungi, which is a wood decay fungi called Stearium. it's a kind of dominant fungi during the whole period. And in terms of the environmental factors, we did quantify the temperature, humidity, total particulate matters, which are considered as physical stressors, kind of, and the other factors we collected here are from the monitoring station, not from the exposometer. So also those three are from the exposometers. And then we did an interomics between the external exposome and internal multi-omics. Those are the three factors we determined in the external exposome, and those are the omics that we did analyze for the internal omics. And we can see that there are like nearly 9,000 significant correlation among all the omics that we quantified here. It's just a brief example of the exposome versus gut microbiome. And we found that interesting, there are like 6 kind of gut bacteria have interesting pattern. They are all positively correlated with the chemical which are shown here, but negatively correlated with the biological stressor in the exposomes. And the last one called roseberry, this kind of bacteria have a totally different pattern with rose 5, like opposite pattern. Here's the example for the metabolome versus exposomes, and we also did the pathway analysis by the significant high degree metabolites. And the result shows that there are very various kind of metabolism pathway are correlated with the exposomes. And majorly, they are related to the liver and kidney functions. The last part is to correlate the cytokines and blood tests with the exposomes.
Can you move to the conclusion? Sorry, can you move to
yes, sure. And the conclusion is we just show that the agrochemical and the fungi are dominating exposome, and 6 kind of gut bacteria is maybe have essential roles. And from the proteomics and the cytokine and the blood test, we found that the liver functions, liver and kidney functions are related to the exposome, responding exposome. And most of the high degree stressors in the exposome are agrochemicals, PPCPs, flame retardant, and certain fungi, plants, and algae. Okay, thank you for your time.
Thank you for your impressive presentation with the analysis of thousands of biological or chemical factors. There are no questions from the audience. I have a question. So, in summary, the different biological or chemical factors are clustered in groups of environmental stressors. In summary, how many of these key factors are the most important one? I don't know if you did a sort of a principal component analysis or that. So which are how many are the most important groups of factors influencing the exposure?
Yes, so I don't have an exact number for you, but if you are interested in the group, we found that a certain kind of agrochemical, most of them are pesticides, and the PPCP and the flame retardant are the ones that are highly correlated with the exposome in terms of the chemical stressors. For the biological one, most of them are fungi, followed by the plants and the algae. And some of them are known pathogens and the virus, yeah.
Okay, so thank you very much.